Hi, welcome everyone. Thank you for joining today's ASBMR Member Spotlight. It's a part of the bi-weekly series that is brought to you by the ASBMR Diversity, Equity, and Inclusion Committee. The series aims to highlight the research of our ASBMR members within a collaborative setting and aims in particular to shine a light on the research being done by ASBMR members belonging to groups historically underrepresented in biomedical research. Today's spotlight will feature two ASBMR members. First, we have Dr. Peggy Cawthon, Senior Scientist at California Pacific Medical Center Research Institute, who will be presenting on the role of muscle mass fails, fracture, and bone strength analysis from the MrOS study. Second, we will have Quamarul Hassan, Associate Professor at the University of Alabama, Birmingham, who will be presenting MicroRNA-Chromatin Crosstalks is essential for bone mass maintenance. As a quick reminder, each presentation will take approximately 15 to 20 minutes, and following each presentation, there will be opportunity for Q&A and discussion with the audience. As a reminder, please try to keep yourself muted throughout the meeting, and you are welcome to unmute yourself for questions and comments following each presentation. So with that, I'd like to invite Peggy to introduce herself and begin her presentation.
Hi, thanks for the opportunity to be here and share our work. It's really fun to have a chance to talk about this work that we've been doing. Let's see, let me go ahead and share the screen and make sure that this works for the slideshow. I've had some challenges with that. All right, can you see this as the slideshow screen?
Yep, we can see it.
Terrific. All right, so as Lauren said, today I'll be talking about the role of muscle mass in falls, fractures, and bone strength. And this will highlight work that we've done in the MrOS study. And I've shown bits and pieces of this at different ASBMR meetings, but never as a whole story. So it's nice to be able to put this together in sort of a cohesive package. All right, so just some background so that we all start on the same page. So one question is, well, why should muscle influence fracture risk? And we know that bone remodels in response to force, such as gravity or ground forces from walking and jumping, or the force that muscles directly exert on the bone. And with greater muscle force and ideally muscle mass, that should translate into stronger bones and reduce fractures. We also theorize that muscle should influence fall risk. We know that most hip fractures are the result of a fall, and that with greater muscle strength and force, you should have a reduction in falls and less fractures. And in fact, this is shown in our work and others, is that poor physical performance is strongly related to fracture risk over time. And so this little schematic on the bottom of the slide here shows that muscle should impact fracture through two pathways, again, through bone strength and quality, and then through falls. So what about sarcopenia, which is conceptualized as the loss of muscle mass and function over time and fracture risk? So there have been at least two meta-analyses of sarcopenia and its association with fractures, but both focused on composite definitions of sarcopenia that incorporate both measures or approximations of muscle size and also measures of physical performance. And one of these two meta-analyses included only two studies. So, the results may have to be taken with a grain of salt given the smaller sample size. Here is the larger of the two meta-analyses that looked at a variety of the different sarcopenia definitions and their associations with fracture. The groupings here on the left-hand side, the EWGSOP, that stands for the European Working Group for Sarcopenia in Older Persons. It's one of the competing definitions. The AWGS is the Asian Working Group for Sarcopenia definition. And then the last one is the Newman definition that was published several years ago now. And overall, there's a suggestion of an association between sarcopenia, these summary measures, and fracture risk. But one of the challenges is it's hard to tease out what component of the effect is due to muscle mass per se or to measures of physical performance. And that wasn't really addressed in this previous work. We know that lean mass from DEXA, which is not a direct measure of muscle, it's an approximation of muscle that includes all the non-fat, non-bone tissue in the body. Often that's restricted just to the arms and legs. While muscle is a part of lean mass is not completely inclusive of, or lean mass does not includes things that aren't muscle as well. And work from us and others has shown largely no effect or no association between DEXA lean mass and fracture risk in either men or women, especially once bone mineral density is taken into account. And as a reminder, lean mass is not muscle mass. So even though these results show that lean mass by DEXA isn't related to a fracture, because lean mass doesn't directly measure muscle, we still don't really know what the association between muscle and fracture is. Here's a nice paper from Nick Harvey's group that has looked at different components of sarcopenia and fracture risk. And here are the 4 components they looked at: time to complete 5 stands from a chair, walking speed, grip strength, and appendicular lean mass divided by height. Again, this is lean mass from DEXA. And when you adjust, when you have unadjusted results, or results just adjusted for age, each of these factors, time to rise from a chair, walking speed, and grip strength, are associated with fracture risk. So is lean mass. But when you further adjust for history of falls, the WHO fracture probability without BMD, WHO fracture probability with BMD, or BMD itself, the associations between the physical performance and strength measures remain essentially unchanged by adjustment. But when the lean mass measure is adjusted by BMD, the association is completely attenuated, and lean mass is no longer associated with fracture risk. And this suggests that lean mass may be, by DEXA, may be a proxy for body size or bone mineral density, and that once you account for bone mineral density itself, there is no independent relationship between lean mass and fracture. So given the state of the literature, we thought it would be a really good idea to look at a new method to measure muscle mass. As opposed to DEXA, this new measure measures muscle mass directly. It's based on an isotope dilution method using deuterated creatine, and I'll explain how this measure works. So to understand how deuterated creatine works, we first need to know some facts about creatine biology. So nearly all the creatine in the body is found in muscle, more than 98%, and it's found at a relatively constant concentration in muscle. So if you're able to figure out how much creatine in the body, then you should be able to estimate total body muscle mass, as these are directly proportional. We also know that creatine is converted to creatinine at a constant rate by a non-enzymatic reaction. One way to figure out how much creatine you have in your body, or your creatine pool size, is to collect a 24-hour urine where all the creatinine in your body is excreted. However, doing a 24-hour urine collection is problematic, particularly in older people who are most interested in studying. There are issues with dietary control, timing issues. You really have to have it exactly 24 hours. And then older adults with incontinence or incomplete bladder emptying or renal problems can cause problems with this measure, because you need to have all of the urine that's collected in a day. So this deuterated creatine dilution protocol overcomes some of those problems. And let me just first explain how it happens operationally and then describe how the calculations occur. So operationally, what happens is on day 0, participants take a 30 milligram dose of labeled creatine. So here, creatine is labeled with deuterium. Deuterium is a stable isotope of hydrogen. It's non-radioactive, completely safe. From day 1, they take the dose. The labeled creatine gets distributed throughout their body. In the interim, in the muscle, the unlabeled creatine that is just there naturally is converted to unlabeled creatinine. And the D3 creatine that the person took is also converted to labeled creatinine in the muscle. So day 1 or day 0, they take the dose, and then 3 to 6 days later, when there's isotopic steady state and the label has fully diluted throughout the body, we take a single spot urine sample. The urine sample is fasting and is in the morning, just to control for any diurnal variations and to make the fasting collection easier. It's not the first morning void, so we try to get a dose that will be not influenced by diet. And from this, we're able to determine muscle mass. So if you imagine this guy on the left, he's got more muscle than the guy on the right. And the guy on the left will have, because he has more muscle, he'll have a higher creatine pool size. So relatively more of his, the creatine in his body would be unlabeled compared to the labeled dose he took. So his ratio of labeled creatinine that he excretes to total creatinine will be lower. Now, this guy on the right is a bit of a wimp. He has less muscle mass, so he'll have a lower creatine pool size. And relative to his creatine pool size, relatively more of the creatine will be labeled by deuterium. And so his ratio of labeled creatinine that he excretes will be higher to total creatinine. And from these measures we take in the urine, that is the ratio of labeled to unlabeled creatinine, we use an algorithm to calculate muscle mass. So we've done that in one of our studies, the large multi-center observational study, MrOS, which is a cohort study that started in 2000 by recruiting about 6,000 participants. At the year 14 follow-up visit, we repeated a bunch of measures in about 1,400 older men whose mean age was 84 at enrollment in this visit. And we did a whole bunch of things to these people. They had muscle mass by D3 dilution assessed. They had DEXA to collect lean mass and a hip BMD. We also calculated Frack's risk of fractures, measured physical performance with chair stands and walking speed, completed both high resolution peripheral computed tomography and also just regular PQCT, and then have collected incident fractures and falls. Fractures were medically adjudicated centrally. Now, when we looked at characteristics of participants in MrOS by quartiles of D3 muscle mass standardized to body weight, those who had the lowest amount of muscle for their body size were older, they tended to weigh more, they had a much higher risk of hip and non-spine and major osteoporotic fractures than those in the highest group. And they also varied quite significantly on physical performance, where people who were in the lower quartile of muscle mass divided by weight also tended to be slower, have worse performance on chair stands, and be less strong. Here's the relationship between total lean mass by DEXA and D3CR muscle mass. Now, neither of these are standardized to body size. And if we thought that DEXA measured muscle mass, we would expect the correlation to be very high and for these points to lie along the line of identity. And we don't see that. And that's explained by the fact that DEXA, in fact, does not measure muscle mass, it measures lean mass. So here the correlation is modest and the relationship doesn't fall along the line of identity. That suggests that they're measuring different underlying things and that the difference between for the line of identity increases as muscle mass increases. So there's greater overestimation of muscle mass by DEXA, the greater the body size. Now here's the relationship between DEXA BMD and D3CR muscle mass, and there's really no correlation. But you'll see here that there's a demonstration of where the hip fractures occurred in this distribution. So on the vertical axis is muscle mass standardized to weight, and on the horizontal axis is bone mineral density. And as we'd expect, most of the hip fractures occurred in the lowest, say, tertile of bone mineral density. Interestingly, the most number of fractures also occurred in the lower half of those, in the lower half of muscle mass. Here's the relationship between incident hip fractures and muscle mass. You'll see that those men in the lowest quartile of muscle mass had about a 5-fold increased risk of hip fracture compared to those in the lowest quartile. This is a really elevated risk, and this is after accounting for age, Frack's probability of fracture, and bone mineral density. They also had an increased risk of major osteoporotic fractures. Here's the survival probabilities of being fracture-free. Men in the red line, that's the people who had the lowest muscle mass, had the most decline in their likelihood of being free of fracture. And this translates to a hazard ratio of major osteoporotic fractures about 2.5 times that for those in the lowest quartile of muscle mass compared to those in the highest. Now, in comparison, there was no significant association between appendicular lean mass standardized to height for major osteoporotic fractures or for hip fractures. So overall, for this bit, we see that D3CR muscle mass is strongly related to fracture, and it's not explained by age, BMD, fall history, or Frack's probability. And I didn't show these data, but it's partially but not completely attenuated by performance measures. We don't know about how this might reclassify people's risk versus Frack's, but those are analyses we're working on now. All right, now let me just show some early data about the relationship between D3CR muscle mass and bone quality. So by bone quality, I mean measures of integral BMD from the HRPQ-CT scans we have, and then finite element analysis measures of failure load also from the HRPQ-CT measures. These measures of bone quality were measured at the same time as the D3CR muscle mass in MrOS, and again about 1.5 years later in about 250 men. And we measured these measures of bone quality by HRPQ-CT at both the distal tibia, which is obviously weight bearing, and the distal radius, which is not. So here are relationships. We just did simple partial correlations where we adjusted for height and weight to remove the potential confounding association of muscle mass. And we'll see that D3CR muscle mass was moderately correlated with integral BMD and failure load at both the distal radius and tibia, but was not significantly associated with aerial BMD from DEXA. DEXA lean mass, on the other hand, was only associated with failure load, and there was no obvious relationship with integral BMD. We also looked at change in these measures of BMD over time and change in failure load over time by quartile of muscle mass at baseline. And you'll see that those in the darker shade here had greater loss of these measures of bone mineral density. This is about a 2% loss over 1.5 years of BMD at the trabecular BMD, cortical BMD, and failure load. Total BMD, trabecular BMD, cortical BMD, and failure load across both the distal radius, distal tibia, and there was no significant association between these measures and aerial BMD by DEXA. So again, men with low muscle mass at baseline had greater declines in these measures, both at the distal tibia and distal radius. However, when we looked by categories of weight, there was largely no association between the baseline weight and change in these measures of BMD or failure load over time. Only suggestion of trabecular BMD change and weight categories. So those data suggest that weight isn't really important for identifying those who might lose BMD, but muscle mass per se may be. And then finally, just one last piece of the puzzle. What about the relationship between muscle mass and falls? So self-reported falls and related fall injuries were assessed in MrOS by querying participants every 4 months by a mailed questionnaire for about 1.5 years after the muscle mass was measured. And again, this is in that same group of 1,400 men. And we've shown a couple years ago now in a paper that we published in Journals of Gerontology that in particular, muscle mass was strongly related to serious injurious falls, so that those who had an injurious fall those in the lowest quartile of muscle mass were about 2.5 times more likely to have an injurious fall than those who are in the higher quartile. And this was robust to adjustment for a variety of confounding factors such as age and body size and physical performance. So overall, in conclusion, we see that this measure of muscle mass is a novel and independent risk factor for hip and other fractures in older men. And we think that our data show that D3-CR muscle mass is related to fractures both by its effect on bone quality and also on its effect on falls. And it suggests that this muscle mass affects both pathways to fracture. We think this also suggests that the role of muscle mass in health in general may be underappreciated. However, I note that there's more work that needs to be done. MrOS is a cohort of only men, who are mostly white, and how this measure works in women and other race and ethnic groups is not clear. And that's really what in the next step happens. I think further down the road, perhaps in 5 or 10 years when we get much more data, we would also be able to determine whether or not this measure of muscle mass could be a simple measure of sarcopenia, a simple integrated measure of sarcopenia for use in the future. I'd like to acknowledge the other MrOS investigators who've collaborated in this work, in particular, the collaborators who've developed the D3CR protocol, who are Bill Evans and Mark Hallerstein at Berkeley, and the data analysts who did many of the analyses for this project. Thanks for your time, and I'm happy to answer any questions that you have.
Hi, Peggy. Nice presentation.
Thanks.
So I had a question about your D3CR.
Mm-hmm.
Have you considered, so we know that there's a lot more creatine in some of your fast twitch fibers versus your slow twitch.
Mm-hmm.
So if we think about age-related changes in muscle mass, a lot of times you'll lose a lot of your your fast twitch fibers, which then lead to an inability to catch yourself if you're falling.
Yeah.
So you cannot right yourself if you start to stumble, and then that leads to a fall. In addition to that, when you think about the loads that are placed on the bone as a result of, for example, fast twitch contraction versus a slow, perhaps the loss of these fast twitch fibers would lead to loss of strain on the bone with contraction, and that could lead to a decline in bone mass.
Yeah.
So I'm just wondering, with your method, are you able to determine, so we only have a small amount of creatine that's stored in the muscle. So are you able to relate that amount of creatine that you're measuring to total muscle mass? And
that's what we've assumed, that there's a constant concentration of creatine in muscle. There's really not a hugely robust literature about the concentration of creatine across different muscle groups in older adults in particular. We have a new study called the Study of Muscle Mobility and Aging that has just started recruitment, and obviously given COVID, there's sort of some slowdowns with that. But the goal there is to recruit 900 older adults who will have a muscle biopsy, as well as this measure of the D3-CR muscle mass. The subset will also have HRPQ-CT assessments. So we'll be able to look at the distribution, in a cross-sectional area, measure the number of fast versus slow twitch muscles and how that relates to the D3-CR and also bone measures. So while right now we can guess about what direction the associations will be, in 2 to 3 years, we'll actually have data from the SOMA study to be able to answer those questions, because I think those are some important considerations. We just haven't had empirical data to test yet.
Have you considered, with your upcoming study, trying to do a correlation between the D3C and muscle strength?
Yes, and we've done that in MrOS for grip strength, and we have a jumping mechanography, where people jump off a force plate, and it's strongly related to strength and power and force at peak power and all the measures you can derive from the jumping power test.
Okay, thank you.
Thanks.
Hi.
I have a quick question. It's Mike Econs. In terms of costs for this, I mean, if it ever did, you know, if it becomes something that would be very useful for clinical purposes, as you move on, what are the costs?
Peggy, thank you very much for the presentation.
Yep.
Oh.
Yeah, just a quick question about cost. I think you may want to move on to the next speaker, but cost is about $125 a person. That includes the dose and the assay. The D3CR, the D3 creatinine and total creatinine are measured by mass spec. And right now that's just done on a regular mass spec. And so, you know, if this were to scale up, those costs could go down quite a bit because you could get a, you know, streamline a tailor-made mass spec to just do these tests. The nice thing is the urine can be collected and then frozen and then assayed all at once. So as long as you can keep the urine, you know, temperature stable, it can be deployed without the participants having to interact with a big bulky machine. So you can mail the dose to a participant, send them instructions, have them provide a urine sample, mail it back to a central lab in a temperature controlled shipper. And so it allows for some at-home assessment of muscle mass too, which is obviously impossible with MR, CT, or DEXA.
Thanks.
I'll start again if that's okay.
Yeah.
I'm curious, did you look at correlations, sorry, with MrOS, did you also have muscle cross-sectional area measures from either PQCT or HRPQCT? And I assume you probably looked at that. And is it correlated at all with either area or muscle density?
Yes, it's correlated with both.
Okay.
So it's actually more strongly correlated with muscle density than it is with area, but it's correlated. I mean, it's like, I can't remember for sure, but it's something like 0.35 with muscle area and 0.45 with density, somewhere in that ballpark.
Less than the DEXA lean mass correlation then?
That was total lean mass. It's about the same as appendicular lean mass. We're putting that those data together for a paper as well. There was another question in the chat about whether this test is translatable to mice. And the answer is yes, it's been done in both mice and rodents or rats. And so those preliminary data are published. And so I can find the link to those papers, but it's definitely translatable to rodents. Any other questions? Great. Thanks for everyone's attention. I really appreciate it. It was fun to share these data.
Thank you so much, Peggy. Dr. Hassan, please feel free to begin your presentation whenever you're ready.
Thank you so much, Lauren, for your help. And I am also very thankful to ASBMR for this, for sharing my research in the member spotlight platform. So today I am going to talk about two different things. One is microRNA. It's on the forefront of gene expression research. And the second one is the chromatin. And how do they crosstalk, microRNA and chromatin crosstalk in maintaining bone synthesis, bone mass, and bone development. I'm very thankful to NIAMS, NIDCR, NIAD, and then the dental school, School of Dentistry, UAB, because some of my students are supported by the dental school training grant. So I will move my next slide. Hello? Okay. So my name is Quamarul Hassan. I have been graduated in veterinary medicine in India. I have a long-standing interest in genetics and epigenetics of skeletal development maintenance. So then I earned my master's and PhD in India, then moved to United States. I joined Gary Stein's lab, Gary and Jane Leon's lab in 2001. And then I moved to University of Alabama School of Dentistry, Department of Oral and Maxillofacial Surgery. I contributed a lot in bone biology research, mostly on homeobox transcription factors and their codes, and then epigenetic bookmarking and retention of memory bank. My current research is microRNA and chromatin regulatory axis. Several folks works with me, Tanner Godfrey, Benjamin, and Theodore Basby. They already graduated. In the audience, you will see Carrie Smith. He is my new graduate student. Dolores Stacks is a postdoc. And then Yuchuan Chen is also a graduate student in my laboratory. So these are the area we work: DNA methylation, particularly CpG methylation, RNA methylation, either adenosine or cytosine methylation in the RNA also. Then histone modification, chromatin remodeling, microRNA, and long non-coding RNA. We work, mostly our hypothesis is how chromatin remodeling and modification is related to the function of bone synthesis. So you saw here, the chromatin is very close on your left side where the repressor complex PRC2 bound there. On the right side, you will see chromatin is pretty open and accessible for transcription factor or replication factors. This is called open chromatin, and they're ready for the synthesis of the transcription. And there is a complex is called BAP complex, BRG1 or Swiss-Snap complex. So PRC2 complex and Swiss-Snap complex, they have a yin-yang relations. One is a repressor, another one is an activator. So we found that microRNA 23A cluster is balancing between these two clusters to maintain the bone homeostasis, synthesis homeostasis, or the maintenance homeostasis. So if I give you a snapshot of gene expression, so see, this is the typical chromosome, and see some parts is open. This part is open for either transcription factors or transcription or maybe replication. And then if everything works in a very nice way, you will see synthesis of messenger RNA and then protein. The messenger RNA are post-transcriptionally regulated by either microRNA or maybe the modification of the RNA. Now the DNA chain can get easily methylated in the cytosine residues. Then there is the nucleosome structure and the chromatin structure. Their octameric histones are there in that nucleosome structure, and they are getting modified in their N-terminal tails. Now, these are like typical chip sequencing data. You will see the modification is a very activating modification. It's called histone H3 lysine 4 trimethylation. So during the differentiation of osteoblast, you will see day 0, day 7, day 14, and day 21. You will see the histone H3K4 methylation is at the promoter. At the same time, you will see when the gene is getting synthesized, you will see there is another histone modification. It's called H3K36 trimethylation that is in the gene body. And you will see by ATAC sequencing, I give an example of a chromatin remodeling factor. It's called BAP45A. The chromatin is pretty open, accessible chromatin. That's why all the transcription is happening. So this is just like an overall idea how transcription happens, what is the status of the chromatin, how chromatin is getting open, and that's why transcription happens and ultimately helps in the gene expression. Now, we have this hypothesis, and we have this central hypothesis in our laboratory that when progenitor cells is going all the way to differentiated to osteoblast or terminal odontoblast, this is mineralized tissue. So I consider both osteoblast and odontoblast going in the same process. So progenitor cells is there, then pre-osteoblast, and then mature osteoblast or odontoblast, and then finally the terminal osteocytes or the odontoblast. How this happens? There is a typical gene expression program working underlying. So they are mostly the bone and tooth specific genes, but there is a very good email communication between these tooth and bones genes with the signaling. So the signaling works along with the tissue specific gene expression. How that works? So the chromatin of these bone or tooth specific genes are very close in the beginning when they're progenitors, then slowly they're getting open. So this process is called chromatin remodeling and modification. So H3K27 particularly, we are very interested. So see, when they are methylated, chromatin is pretty close. But when they're acetylated, chromatin is pretty open, and the gene synthesis happens when H3K27 gets acetylated. So we call that signaling is working definitely upstream, but signaling is cross-talking with all the bone and tooth specific gene when their chromatin is open. This is also, we call, this is a predetermined genomic accessibility. If osteoblast doesn't have predetermined genomic accessibility, it cannot be differentiated though. So what we found that there is a microRNA cluster. I will go in details a little bit more, but we found in our laboratory, this is a microRNA-23 cluster. So when we study the gain of function of this microRNA, mostly it recruits H3K27 methylation when the chromatins are close. But in the loss of function study, when microRNA-23 cluster loss of function, we saw it's pretty open chromatin. So they mostly help to recruit H3K27 acetylation on this promoter, bone-specific or tooth-specific promoter. So we came to this central hypothesis that microRNA cluster function is linked with an epigenetic homeostasis during bone and tooth formation. So the next one, why we do is this kind of study though? Because clinical and translational significance of my project is the worldwide, there is 8.9 fractures per year, and that results in one fracture in 3 seconds. And the second one is after 50, both men and women, we suffer like 0.5% bone loss in each year. Astronauts even lose more. They lose 1.5% of bone in just 1 month. And see, over 53 million of adults Americans have low bone mass. That's because of chronic diseases like arthritis, osteoporosis, or periodontitis. So we need a novel therapeutic strategy to protect the bone loss. And definitely we need that. That's the primary significance of our project, like why we want to do bone research. The second one, the premise of our research is, only 21 to 25,000 proteins we have. And that is actually 2% of human genome. Now what happens to the rest 98%? That's the non-coding genome. And non-coding genome has a very important role in the maintenance of our chromosome. Now 50% of the BMD loci has already been identified by genome-wide association studies. And only 10%, unfortunately, the genes has been linked to skeletogenesis. So hence, we think that epigenetics has a bigger role or a very important role in osteogenic or odontogenic differentiation or the development. Now, epigenetics is an extra layer of information that is in your genome, but that's not encoded by your DNA, but they're very important in genomic function and specific function, whether the gene will get open or whether the gene will get closed though. Now, among them, this non-coding RNA, microRNA is very key research area. And I think day and day, there are a lot of microRNA research happens in bone formation and bone maintenance. And the second area we do research is chromatin remodeling and modification. That's another area that's in the forefront of bone research and cancer research also. So now, in almost 8-9 years ago, 2010, when I was working in Gary Stein's laboratory, we found and we identify a microRNA cluster that have 3 microRNA in it, 23A, 27A, and 24-2. So we found that this microRNA has a very potent role in osteoblast differentiation, because when we overexpress microRNA 23, there is a delay in osteoblast differentiation. Both alkaline phosphatase staining and bone consortia staining is telling that. And 27A is also delaying, but 23A is more potent and almost blocking the microRNA osteoblast differentiation. So from that, I was very interested in this cluster. So when I became independent and took position in University of Alabama, I just tried to make this microRNA mouse model. The first microRNA mouse model I made just replaced the whole microRNA cluster with puromycin. And then we found that when I delete this microRNA, it's a heterozygous condition. This microRNA deletion forms more bone. But homozygous mouse, in this condition, they die. So I took my second approach and make an anti-microRNA, and that anti-microRNA expression is regulated by tetracycline responsive element. And when I just treat the cells or when I feed the mouse with tetracycline, the ROSA locus at the same time was modified with reverse state activator. So when I feed the mouse or treat the cells with doxycycline, more and more anti-microRNA got synthesized here. And what ultimately happens, this anti-microRNA block the function of the microRNA. And then when we develop the mouse, we found that the heterozygous mouse have even much more bone formation. This is the trabecular bone. But again, when we feed the mouse with too much doxycycline, the mouse die. So that means it is dependent upon the anti-microRNA doses, more and more anti-microRNA, which completely block the microRNA. So microRNA this cluster may be very essential during early phase of development or early phase of growth in mouse development. So then the next approach we took, can I make a tissue-specific mouse? So we just manipulated or we just modified 3 alleles. We just introduced a Cre recombinase with collagen 1A1 promoter. And then second one, we just fluxed the RTTA locus that was in ROSA locus, so introduced CAG promoter. And we just fluxed the stop sign in between CAG promoter and the reverse state activator gene. And then we ultimately have this allele modified. That one has tetracycline responsive element, anti-microRNA, all 3 anti-microRNA over here. And how it works, like in the first things, like 3 recombinase will get synthesized, and then they will take out the stop signals in between CAG and the reverse state activator. And then finally, this reverse state activator will bind along with doxycycline to the TET responsive element, and you will see more and more anti-microRNA, and that anti-microRNA particularly will work in osteoblast cells. So whatever phenotype we will see, that is particularly in osteoblast and very specifically in osteoblast cells. And very interestingly, we found that both cortical bone as well as the trabecular bone is very much increased when we treat or when we feed the mouse with doxycycline. So my conclusion is this microRNA was inhibitory for bone formation. So when we do a complete knockout in osteoblast cells by conditionally taking him out, we found more and more bone formation. As it evidence that you will see the bone volume over tissue volume is highly increased in both male and female mouse. And we then continue to do the bone strength is pretty higher for this mouse. Then we did the RNA sequencing of the doxy-treated versus non-doxy-treated cells from this mouse. And we found mostly there are 3 pathways, extracellular matrix pathway, and the collagen containing extracellular matrix and ossification is very highly regulated by this microRNA. Then also there is very interesting that one of my postdoc, particularly she's in the audience, Dolores, she took the PRX1 key and just take out this microRNA very early during development. And what we found, this mouse does not form the fourth limb, either unilateral or bilateral way. So this study, Dolores, is continuing. This is very interesting. And then another student, he is working on that. Kelly Smith is working on this project. We took this microRNA very early in pre-odontoblast cells, and we found that this microRNA is also very useful or very important for the tooth formation. So this is a video I can share with you. Dolores Stacks is working on this project, that how this. So these these mouse are very hard, and they have very hard time in walking. They're just like they cannot walk. Either their forelimb is actually messed up, or both the limbs is messed up in this mouse model. So my laboratory is very much interested in the chromatin remodeling and modification. So when we analyze very in-depth of the RNA-seq data, we saw there are 277 transcription factors that very much significantly changed during the knockdown of this microRNA. And we found among them, there are 54 chromatin factors is highly also significantly changed, and 39 of them is directly involved in skeletal development. So we revalidated the whole sets of, particularly for particularly for the chromatin remodeling enzymes or chromatin remodeling factors. And we noticed a very interesting thing, that there is a molecule called BAP45A. It's a chromatin remodeling factor. And at the same time, there is a molecule called EGH2. It belongs to PRC2 complex. And we found when this knockdown of this microRNA very much increased the expression of BAP45A, but they very much decrease also the expression of EGH2. So we found a very good correlation that one complex of the chromatin remodeling factors is actually opposing the function of PRC2 complex. That might be the reason for excess or high bone formation. And then we again concluded this way that microRNA-23, a cluster knockdown, robustly upregulated BAP45A member of PBAP complex, but at the same time inhibits the expression of EGH2 from PRC2 complex. So then we just because this mouse has been developed by Crabtree, a Stanford group. So we found that BAP45A, this is my second project. So we just ordered this mouse from Jackson Laboratory. And then when we did a tissue-specific deletion with osteocalcin 3, very much we saw there is a bone formation is inhibited in this mouse model. Both cortical bone is down, and then both trabecular bone is also reduced. You will see the histomorphometry of this mouse. The femur bone formation rate is decreased. But very interesting thing, we found that the bone marrow contains very high amount of fat when we take out this chromatin remodeling factor, particularly in bone cells. So these factors is a part of the BRG family, Swiss snap complex family. So you will see 45A is here. The main function of this complex is to slide the chromatin and make accessible for the transcription factors. So we were very lucky. Our microRNA has a phenotype at the same time our BAP45A or the chromatin remodeling factor has a phenotype too. And now we did the RNA-seq for this BAP45A, and we found a lot of changes in the expression of the genes. I'm not going too much details on that, but what we conclude from there that there are two axes is being regulated by this BAP45A. One of the axes is RUNX2 and KLF4, and the second axis is RUNX2 and FGF18 or FGFR2 or 3, the receptor axis. So we are working on these two axes too. Now the final slides of my talk today. I didn't go all the way to all my data. I just gave some kind of overview of some of the data. But how we could go from my basic research to translational research though. So simply we found this anti-microRNA are very potent candidate for bone formation or bone synthesis when we take it out. So we have this anti-microRNA. Now we can deliver, and one of the OMS resident is working on that. If we do fracture in microRNA wild type and then dogs treated versus dogs non-treated mouse model to see whether they have a more regenerative capability. Another very interesting thing I saw is, this is a chromatin remodeling complex. This is the normal expression, like BAP45A is always higher, and BAP53A is also always higher, and BRD7 is also higher. But when we took out BAP45A, we completely found a switching of this complex from BAP53A to 53B or BRD7 to BRD9. So one very interesting thing I came across with a Nature Review drug discovery that bromodomains, so BRD7 and BRD9, they're all bromodomain. So they are the new class of drug targets for cancer drug development. So I'm thinking that maybe some small molecule in this way, if we can prevent the switching, then we can also prevent the bone loss, or we can maintain the bone mass for a longer period of time. So this way, either anti-microRNA, or maybe I can, if I just inhibit the switching between this substrate switching, maybe we will be winner and we can regenerate bone during osteoporosis, or we can maintain the bone during osteoporosis, or we can regenerate the bone during fracture. With all this, I'm very thankful to Lauren and the ASBMR, and I am very thankful to all my funding, my family, and the New York School of Dentistry. And thank you, everybody. If you have, I'm ready to have any questions.
Hi, Dr. Hassan. Thanks for the presentation. So you had mentioned on one of your slides about the increase in bone loss. I think you said it was like 1.5% in astronauts versus, people that are on Earth that lose a 0.5% each year. You had also had your knockdown of your MicroRNA-23A. I was just wondering, have you considered doing any studies looking at with your knockdown model and looking at hind limb suspension to kind of translate it to see what would happen to mimic space flight?
I didn't do that, but this is a very interesting area. Maybe I need to collaborate with somebody who can help me. So if you are doing that kind of study, I'm very, very happy to collaborate with you.
NASA may be interested in this type of model.
Yeah. Thank you.
So it looks like there might be someone, Eileen from the chat, asked a question. Is the PRRX1 CRI microRNA knockout specific for forelimbs? If so, any idea why?
I'm not sure, because PRX1 CRI is very specific for limb bud mesenchymal cells. There are two pathways going on that. There are Hox pathway, and then there is another protein is called, there is another transcription factors. They are also, let me find that slide. Probably I'll find. Yeah, so there are another transcription factors on that pathway. It is called TBX5 and TBX4. So TBX4 forms the hind limb, at the same time TBX5 forms the forelimb. So we are making more and more mouse right now with PRX1 and we'll go directly to synthesize, isolate the limb board, and we'll do RNA-seq from that. If we find a candidate gene on that pathway that might be very restricted to the forelimb, or that might be very restricted to the hind limb, then we might answer that question in a very better way though. Right now we don't have any clue though. If so, any idea why the not just the forelimbs? So sometimes we saw like only on the forelimbs. We also saw it actually messed up both forelimbs and hind limbs too. So there is another paper I came across recently. There is a HOX 13 versus HOX 11. They are very much responsible for development of the forelimbs and hind limbs. But sometimes because of the patterning defects or because of the patterning, one is taking over the other one. So we found that paper that HOX11 is actually taking over HOX13. So they might switch the functional aspect of the development. I am not sure right now. Hello?
It looks like Eileen answered in the chat. Thanks, very interesting for addressing your question.
Yeah.
Well, are there any last questions for Dr. Hassan before we wrap up? Okay, great. Well, thank you both, Drs. Hassan and Cawthon. Those were very interesting presentations and great questions by the audience. Just as a reminder to everyone in attendance, the last member spotlight of 2020 will be 2 Thursdays from now, December 17th at 3 p.m. Eastern Time. And please be on the lookout for the schedule for the 2021 member spotlights, which will be coming soon. Thanks again, everyone, for your time, and I hope you have a great rest of your days.
Thank you so much, Lauren. Thank you, everybody.
Thank you.